Hi Year 10. This week's task for biology is all about cell transport. Last week you looked at the parts of cells and cell structure and being able to view cells with a microscope and all the skills that entails. This week we're going to carry on with Unit 1 revision and looking at the required practicals, and there's a hideous image on the screen there of 3 eggs, one of which has been left by itself, that's in the minnow. One has been put in one type of solution and the other one at the far end has been put in a different type of solution. And what you can see there is that one of them remains unchanged, one looks like it's blown up like a balloon, and the other one has shrunk right down. And today's work is recapping the science that explains that. So hopefully when we think back to that image over the lesson, you might have some ideas about how we could explain that. So just to recap before we carry on with cell transport, just pause the video when I finish speaking and give yourself a chance to revisit what we looked at last week by reminding yourself which of the cell parts we find in animal and plant cells answer all these questions. So which cell part contains DNA, which cell part there carries out aerobic respiration. So pause the video and when you restart, we'll go through the answers. Just jot down your answers. Okay, hopefully you identified these answers, that the nucleus contains DNA, mitochondria is where aerobic respiration happens, the cell membrane controls the movement of substances in and out, so that's going to be very important for today's lesson. And there are 3 parts not found in animal cells but are in plant cells. Hopefully you found at least one of those: chloroplast, vacuole and cell wall, and protein synthesis happens in the ribosomes. Hopefully you did well there. If you didn't, you might just want to run back over that either in Seneca or BBC Bite Size. So today's work is all about cell transport, and that doesn't mean moving cells around, but moving things in and out of cells. So on the left is an animal cell, on the right is a plant cell, and substances needs to move in and out of each type of cell all the time. So we're thinking about things like oxygen needing to get in and out, depending on which type of cell it is that you're talking about, carbon dioxide, same, and also depending on what time of day it is for the plant cell, might need to go in or out. Glucose and minerals, all those things need to move in and out of the cells all the time so that the cell can function and the organism can continue to live. So there are, and you've learnt this in year 9, we've revisited it at the beginning of year 10, there are 3 ways that substances can move in and out of cells. So 3 processes by which substances move in and out of cells. So the first one is diffusion. Hopefully you recognise that word. The second one, osmosis, and I know you'll recognise that word because I'm obsessed with it. And the third one is active transport. You need to recognise those 3 processes, be able to describe them, hopefully know examples of them as well, and be able to apply your understanding of those 3 processes to different practical data and scenarios. So diffusion is where substances move from a high concentration to a low concentration, and you can pause the video at any point here if you want to make yourself some notes to go with last week's revision materials. A good example of diffusion is when oxygen diffuses into red blood cells from the alveoli in animals. Or I should say animals with lungs anyway, so that wouldn't happen in fish. Still going to red blood cells, but not through the lungs. So oxygen is moving from a high concentration of oxygen in the lungs to where it's a lower concentration, and that's in the red blood cells. Osmosis is the diffusion of water. So it's something moving from a high concentration to a low concentration, but specifically it's water. And this is a really important definition. It's the diffusion of water from a dilute solution to a concentrated solution through a partially permeable membrane. Lots of key words in there that we need to kind of unpick to be able to understand what that definition is telling us. The first one is dilute. So dilute means something that's very watery. So when you make yourself a glass of cordial, you dilute it by adding water. So a dilute solution is one where there's lots of water. A concentrated solution is the opposite. Concentrated solution is like the Vimto before you've diluted it. So concentrated just simply means where there's not very much water. So the movement or the diffusion of water from where there's lots of water to where there's little water, and that water is going through this funny phrase here, a partially permeable membrane. You'll recognise membrane because there is a membrane on the outside of cells. And partially means not fully. Permeable means that it lets stuff in and out. So it's through a membrane, like on the outside of a cell, that lets some stuff through, but not others. So it lets small molecules like water through, small molecules like salt and sugar through, but not big molecules. So that's just a really long-winded way of saying that water is moving into or out of a cell from where there's lots of water to where there's very little water. And the example of that is water getting into the root hair cells of plants and water going into our bloodstream in the large intestine. Active transport is different to both of those two processes because it's the movement of substance from a low concentration to a high concentration. So it's going from where there's very little of the substance to where there's already lots of the substance. And that needs energy because it's like trying to shove an extra pair of socks into a drawer of socks that's already full. It's going to take energy to try and get it into there. And that energy comes from the mitochondria who are carrying out aerobic respiration. So cells that do active transport will have lots of those mitochondria. Good examples of these that you do need to remember, particularly the first one, is that nitrates that plants need for growth, nitrates get into the root hair cells of plants by active transport. So the concentration of nitrate is low in the soil and high in the root hair cells of plants, but the plants need it for growth, so they absorb it using energy by active transport. The other example is glucose moving into our bloodstream at the end of the small intestine. At the top of the small intestine, straight after the stomach, glucose will move into the blood by diffusion, but at the end of the small intestine, it happens by active transport. Okay? If you want to pause and make yourself some notes on that, you can do. But I'm going to just check now that you've understood those three processes by simply asking you to write down the answer to each of these. So for each of these four statements, it's describing one type of cell transport. Actually, one of them is describing two, but just write down your best answers to those, decide whether it's osmosis, active transport, or diffusion, and jot those down after you've paused the video, and then we'll check your answers. So the first one is osmosis because it's talking about water and this membrane, so that must be osmosis. The movement of substance from a low to a high concentration, that's active transport. The movement of substance from high to low is diffusion. If that said the movement of water, it'd be describing osmosis. And the last one, the one that needs energy, the only one that needs energy, is active transport. If you want a little bit more of a challenge, you could use this structure to draw a Venn diagram to compare the three types of cell transport. So the overlapping areas here, that one being where there are things in common between diffusion and active transport. And there are some, on the next slide, there's some labels that you could add to that Venn diagram. So if you want to pause and do that, you can, but that's a bit of a challenge task. The answers will pop up in a moment for those people who want to do it. But do pause the video if you're planning to. Okay, so that the answers are there for you. I've just used the initials. Hopefully that's clear. If it's not, please do get back in touch with me via ePraise. And we'll move on now to think about one of those types of cell transport in quite a lot of detail. The second required practical that you did is described here on the screen. Hopefully you remember it. It's a little bit of a faffy one, but you get really good results for this. And then we all draw a beautiful graph and we marvel at our lines of best fit. And when we do it, it really makes sense. But you've not done it for about 12 months, if not longer, so it's worth taking the time now to recap it. So this is a required practical where we're investigating how the concentration of a solution affects the change in mass of a potato. So we use potato chips. Sometimes you could use, if you wanted to spend a bit more money, you could do it on carrot, you could do it on apple, you could do it on little worms if you wanted to, but we use potato because they're cheap and easy to get hold of. So I'm just going to describe the practical, and then I'll show you the method for it. So we use potato chips, and we cut them all into similar sizes by cutting the ends off, and we put them in a range of different salt solutions. So some very watery solutions and some quite concentrated, really salty solutions. And we leave them in there for a period of time, and we measure afterwards how much they've changed in mass. Some of the potato chips will go up in mass and some will go down. Now, this practical, it's not the salt that's moving, it's water that is moving in and out of those potato cells. So it's water going from where there's lots of water to where there's very little water. So it's a practical that's all about osmosis. And the whole point of us doing it is to be able to work out how salty the potato is to begin with. So working out the concentration of salt inside the potato. Now, I'll just tell you here that sometimes the exam board will describe this practical, but they won't talk about potato. They'll give you the example of a carrot or an apple, but the science is the same. Even if it's a worm, the science is the same. It's all about osmosis. Now, if you look here, we're trying to work out the change in mass of potato, which means we need to measure their mass at the beginning. We measure the mass then at the end, and work out how much they've changed. So here's a written method for the practical, which is very similar to the one that you will have carried out in class. So pause that and read through it, or if you prefer, you can look at the kind of picture version of it on the next slide. There you go. This has got an extra step in that sometimes the exam board will include. I never do it because it just takes us too long in a lesson, but you can also, as well as measuring the starting mass, oh God, what's that? Sorry. As well as doing the starting mass, you can also measure the length of the potato at the start and the length of the potato at the end. But either pause here or go back and read the written method so that you know what the practical is actually involving. Hopefully it'll jog your memory. If it doesn't and you don't remember it in very much detail at all, there's a link in the ePraise notes to a different video on YouTube that describes that and shows you physically, you know, with a man and actually demonstrating how they carry out the practical. Okay, so this is how it might look. As you can see here, this is an exam question from the exam board, and they've used carrot in these 1, 2, 3, 4, 5 test tubes or boiling tubes. They've used carrot. Doesn't matter, it's exactly the same practical, you're just thinking carrot instead of potato. And here's the sort of results that we would get from this practical. So they've used 5 different concentrations of sugar solution. So we use salt, they've used sugar, still the same. So they've got a 0 concentration, which is completely water, slightly more sugar again, more sugar again, and then a really, really sugary one at the end. And what they've worked out is the percentage change in mass. So we work out the change in mass first by doing the start mass and the end mass, and then you can calculate the percentage change in mass. And that's quite important because you probably remember doing this practical, really hard to get them all exactly the same mass to begin with. So you might have one that's 2 grams, 2.1 grams, 2.05 grams, 1.999 grams. Very difficult to get them all exactly the same starting mass. So if we work out the percentage change in mass at the end, it means we can compare them more easily because they won't all be the same starting mass. To work out the percentage change in mass, oh crikey, sorry, you do this, the change in mass divided by the starting mass times by 100. That's a really important calculation. They will ask you somewhere to work out a change in mass, percentage change. It might not be to do with this practical, it could be anywhere, but the change divided by the start times by 100 gives you percentage change. So if you look here, these numbers about the percentage change in mass correspond to these 5 different test tubes full of carrot. The first one here at 0% concentration, 0 concentration I should say, that one, that's gone up by 24%. So that's gained mass. The next one has gone up 12, so it's gone up, but not as much. The third one has gone up as well, so it's gone up by only 1, but it has gone up. The next two, so in a stronger sugar solution, this one has gone down and that one's gone down too, but by a greater percentage. So these three have gone heavier, these two have got lighter. And why is that? Remembering that I said it's a practical all about osmosis and that water is moving. These three, water has gone into the potato cells from the solution. In these two then, if they've gone down in mass, what must have happened? They've lost water. Water has gone out of the potato cells. So in what situation then would water not be going in or out? Well, somewhere between these two concentrations. This one has got heavier, this one has got lighter. So water's gone in that, water's come out of that. So somewhere in between those two concentrations, if you investigated it, you would find that water isn't going in or out. And you can plot that on a graph. You can plot it on a graph so that you don't just know that it's between 0.4 and 0.6 moles per decimeter cubed where no water's moving, but actually much more accurate than that. We can predict whether it's 0.5 or 0.52 by drawing a graph. So you would have axes. On the y-axis, you've got to have a positive and a negative scale. So 0, change in mass, would be here. These have gone up in mass and these have gone down in mass. And on the x-axis, you would have your independent variable, and that's the salt or the sugar solution, whichever it is that you're looking at. And then you'd plot your points. So the first one at 0% salt solution, that one went up by the most, all the way up here. So you draw a little X, having done a scale and all that jazz with your numbers. The next one went up as well, but not as much. The next one went up, but only a tiny bit. The next one went down, and the next one went down even further. Then, as you know, you would want to draw a lovely line of best fit there, so connecting as many as possible, but not doing it dot to dot. Line of best fit, something like that. And if you remember, we said that the ones at the top here, they've got heavier because water has moved into the cells. The ones at the bottom here, the water has moved out of the cells. So where is there no movement? And it's there where the x-axis is being crossed. So the line of best fit is crossing the x-axis, and it's whatever number that equates to. So if you had a real graph here, it'd be 0, 0.2, 0.4, 0.6. So you'd be able to work out using the little squares on your graph paper whether that was 0.5 or 0.52 or 0.54 or whatever. Okay, so what you can tell from that picture then is, or sorry, I should say from that graph, is what the concentration of salt is in the potato. And that is commonly quizzed on exam questions, okay? That your understanding is there to be able to plot the graph, because that's a difficult skill anyway, and that your understanding of why you're carrying out this practical is there as well. Okay. So just now we've gone through that quite quickly, you might want to just pause the video and make yourself some more notes about that practical by identifying the 3 types of variable: the independent, dependent and control. Independent's what you change, dependent's what you measure, control's what you keep the same for validity. And then have a think about why we carry out some of these steps. So why do we weigh the potatoes, blot them? That just means dab them with paper or something. Why do we blot them before we weigh them? Why do we calculate percentage change and how so? And then how do we use that graph and what we're using that graph for? So just make yourself some notes after you've paused the video, and then I'll show you the answers. Okay, here are the answers. So you just pause the video again and check that you've got something similar to what I've written there. And we're now going to take a look at some of the exam questions that that I'm setting you as part of your revision and your assessment this week. So the exam questions will start with this question, which is exactly the same practical. It's asking you to do a bit of a calculation here, and then some written questions, which hopefully the video will have given you enough guidance to be able to crack on with. We are going to look at this one, which is the end of the second question, and it's all about that carrot practical that I showed you before. So the one with all the test tubes. This question wants you to explain why the mass of the carrot in the 0.6 mole per decimeter cubed sugar solution changed. So explain is our command word, and that means you've got to give some scientific reasons. And if we just highlight the key information in the question, it wants us to talk about why the mass of the carrot in the 0.6 molar per decimeter cubed solution has changed. So this is a 4-mark question, which means you've got to make 4 clear points or use 4 distinct keywords as part of your explanation. So what you might want to say first of all is whether it's increased or decreased. So look back at the table of information, or look back at your graph you've drawn, and have a look for the 0.6 molar one. So that's the 1, 2, 3, that's the 4th one that they've investigated. Have a look. They've said it's changed. They want you to be able to interpret that data. Has it increased or decreased in mass? So that's your first starting point, because they've not told you, so you should show that you know. Then you're giving the why. So you're thinking about these things. You need to say that it's water, that it's moving. You need to say how that water that it's moving, which process, so name the process. And you need to talk about cells. So it's water going into cells or out of cells. It's not going into the potato. And make sure you mention, just to round it off, the partially permeable membrane. And if you can, you should give a reason why the water has gone in the direction that you've said. So has it gone in or out of the cells, and why? By thinking about where is the solution most dilute. Is it more dilute on the outside of the cells or more dilute on the inside of the cells? Okay. One of the beginning parts of the third question isn't just about osmosis. This question, this is asking about cell transport in general, and it gives you a bit of information at the top and says that the table shows the concentration of 4 ions. So an ion just means a charged particle from chemistry, outside cells and inside cells. So it's got a list of the ions, the concentration outside, and the concentration inside the cells. And the question says, use the information from the table above, which tells you the answer is here. No, you don't need to guess. Use the table from above to complete the following sentences. Sodium ions will move into the cells by the process of what? And then potassium ions will move into the cells by the process of what? So there are 3 processes that we learned about that move things in and out of cells: diffusion, osmosis, and active transport. And you need to have a think to yourself, first of all, which one is it definitely not? Well, it's definitely not osmosis, because osmosis is the movement of water, and the first one is about sodium and the second one's about potassium. So it's definitely not osmosis, so we've narrowed it down to either diffusion or active transport. And then you need to follow their advice and use the information from the table. So you've got the concentration outside and the concentration inside the cells, and both statements are about them going into the cells. And then you've got sodium on one line and potassium on the other. So you need to start with sodium. So the concentration outside is 140. The concentration inside is just 9. So that's going from a high concentration to a low concentration, whereas the potassium underneath is going from a low concentration of 7 to a much higher concentration of 138 in the cells. So from a low to a high concentration. So then you just need to think back to your definitions of those two key terms that we've narrowed it down to and decide which is which. The rest of the questions, I hope that the video gives you enough help and guidance to be able to have a go at. So you can rewind the video and go back to the graph, and you can go back to the explanation of what osmosis, diffusion and active transport are at any point. There's also a lesson on osmosis in more detail on the Pearson website on YouTube, and I'll send you the link for that just in case you want more information. But if you do look at these questions and you're really struggling, get in touch with me please on ePraise, and I'll be happy to help. Okay, thanks Year 10, see you soon.